morning, everyone. Thank you for joining us. It's my pleasure to introduce you today, our speakers of today seminar. Ramos, he's one of our ESIIL postdoc, and he is an expert in quantitative ecology, studying terrestrial and aquatic microbiomes. His work focused on species interactions and mutualism, how change in the microbiome community structure affects community function, applying microbial ecology to ecosystem management practices, and modeling patterns of species diversity at geographic scales. A quantitative approach to research appeals to him because it leverages novel analytic approach and allows for a high multidisciplinary collaborative approach. In his past work, he had looked at questions of microbial distribution and community structure across stream networks, utilizing techniques for analyzing direct acyclic graphics. Much of his research has also focused on the evolution and persistence of symbiotic relationships. Mycorrhizal fungi are a vital part of the soil community with strong symbiotic relationships with many prior plants. His current research hopes to expand the understanding the spatial patterns of niche characteristics of arbuscular mycorrhizal fungi, utilizing niche modeling techniques. So thank you, and the floor is yours.
Thank you so much.
Yes.
Thank you all for coming. I'm very excited to be able to share some of my past research with you all. So overall, the overall theme of this talk is going to be using quantitative approaches to understand microbial responses to different kinds of environmental change and their ties to like anthropogenic environmental change. There are going to be two broad studies I'll be talking about in this talk. One that's going to be looking at the plant soil feedback, so using kind of like this framework for looking at changes in plant communities and soil communities together to understand their responses to things like land degradation, but also like plant community change. And then I'll also be talking about a project that did looking at microbial community change in an intermittent stream network. So the idea is stream networks that dry periodically, how do the microbes respond to that? And what might we be able to extrapolate from those systems when we're dealing with a warming climate and more streams are becoming these intermittent systems that dry periodically. So that's what I just said, general outline of the talk. And then at the very end, I will talk a little bit about what I hope to do here at ESIIL and how that's going. So starting off, the North American prairie system is if people who work in the system are a little bit befuddled by this consistent problem that keeps coming up, which is that when you do a prairie restoration, most prairie in the United States has been tilled, turned into agriculture. There's very little of the remnant prairie left. So a lot of the work that's done in prairie systems is restoration, bringing back old farm field, bringing them back to prairies. But this work constantly keeps running into this problem where the restorations do not reconstruct the total diversity that you see in the prairies. They're at a lower diversity state, happens very consistently. And people are trying to understand why these restorations often, not every time, but often we'll call it fail to restore the original biodiversity. So one aspect that could be important to explaining this is what's happening in the soil community. So I'm really interested in mutualists. It's kind of a passion of mine, and a very important soil mutualist, particularly in the prairie, are these arbuscular mycorrhizal fungi. So these are fungi that live in the soil. They have no above ground structures. You've never probably seen one because they have no mushrooms. They never produce above ground structures, but they're extremely important to plants. So what the structures they produce are all below ground, and one of the key structures for this group are these arbuscules. So you can see that you have a fungal spore, that is a little round symbol. Fungi create hyphae, which are these thread-like structures. You can think of them as analogs or roots for fungi. They spread throughout the soil, they collect nutrients for the fungi. And in AMF, these hyphae can enter the plant roots. So these are plant cells. they will actually integrate into the root, and then they'll form these structures called arbuscules. These are actually like, they're membrane separated, but they're growing into the actual cell structure. And these are sites of nutrient exchange. So these AM fungi actually will pass on nutrients that they're collecting from the soil to the plant. And AM fungi are obligate neutralists. They don't generate their own carbohydrates. They don't generate their own sugars. They get all that from the plant. So they have to associate with the plant or they can't grow. And they generate these huge benefits to plants because these fungal hyphae grow faster than roots. They're finer than roots. So they can be very effective at acquiring nutrients that would be more difficult for the plant to acquire using a fine root system. So they basically mine minerals from the soil. In the case of AM fungi, it's the most important nutrient is phosphorus for these groups. They're really good at acquiring phosphorus. Plant gives them sugar in exchange, and that's the backbone of this mutualism. So a lot of studies have been done on this plant soil system. We know through past research that most plant species actually associate with AM fungi. They get different levels of benefit, but there's a large number, especially in the prairie. It's more of a mixed story in forests. There's kind of this other whole other group of mutualist fungi called EM fungi that are very important in forests. But in prairie, AM fungi are dominant. And then again, they improve the resource uptake of their host plants. And we have seen that structure of the AM fungal community, particularly diversity, has been shown to have impacts on the functioning of the above ground community. So I want to understand this relationship better. And, oh, sorry, I'm missing a step. We also know that in the other direction, the structure and the traits of the plant community will shape the AM fungal community. They also respond to the plants that are present. A big plant trait that I want to hone in on what I'm going to talk about today is this life history. And life history is kind of like this catch-all term I'm using to talk about these two different life strategies. We have early successional plants, late successional plants. You can think of this as basically R-selected and K-selected, if you guys remember those terms. We have these fast-growing early successionals. They invest a lot of their energy in the above-ground structures, and particularly seeding. So like they tend to be annuals, and they tend to invest heavily into reproduction every year, and that's their life history strategy. And then you have these late successionals. Some annuals, mostly perennials, so mostly plants that come back every year. They invest a lot more in the below ground structures, particularly because those structures in these herbaceous plants are the ones that are perennial. Those are the structures that stay around and they regrow their above ground biomass. So I want to understand how the AM fungal community is shaping these plant communities and how traits like life history are shaping the AM fungal community, and how is that feeding into each other in like a feedback loop. The last thing I really want to look at, because it's an important structure that often gets talked about, but it has not been often rigorously studied, which is the relatedness aspect of the plant community. It's not been studied enough because it's very difficult to study it. You need to really sample across the phylogeny of plants to be able to make meaningful interpretations of your data. So I set out to study a large number of plants across the whole plant phylogeny to try to get at this question of overall plant relatedness. How is this also affecting the relationship? Are more or less related species? Does that help you predict what's going to happen with the soil community? So first research question, how does the AM fungal community respond to plant characteristics? And in my case, in this study, we're going to be talking about plant relatedness and their life history strategy. How do I do this? Well, I did this in a greenhouse experiment. So we took a bunch of different plants from the prairie, we planted them into homogeneous soil communities. So the soil communities all start the same. I'll talk a little bit how we did that in a second. After growing season, we are going to, after growing season, they're growing with these plant communities, or these individual plants, the soil communities will change. This is an established experimental design. I promise you that the soil communities will change, but we will also measure it and we'll see how they change. And then finally, we sequence the soil. That's how we're going to detect if there's been a community change in the soil. So again, I needed a broad selection of host plants to be able to test the phylogenetic effect. So we took 38 different plant species. We tried to spread them across major groups. So the groups that we kind of focused on were lilies, otherwise known in our case as other monocots, so non-grasses, grasses, legumes, milkweeds, where in the prairie people get a lot about milkweeds because of butterflies, monarchs, mints, and asters. So we also wanted to select between early and late successional species to be able to test that effect as well. So these are the plants that this is the plant selection that we did. Ends up being a very big experiment. So this is the actual greenhouse we did it in. A lot of hours standing on concrete slabs and looking at plants. And then in terms of the soil community, we started with a homogeneous soil community. How did we do that? Well, we started with steam sterilized soil. So everything was killed in the soil. And then we took from cultures these 7 AM fungal species. We inoculated from spores. We did this because we know something about these 7 species. They've been used in previous experiments, so we have some prior knowledge of them, and now we have control. We know that nothing else is alive in the soil. We're just looking at the effect of these 7 species, and we can measure the changes in these 7 species. And again, the only structure that really are photogenic in these AM fungal are the spores. So a nice picture of the spores. All right, and the sequencing, the last bit of the methods, we did Illumina next-gen sequencing. We're targeting a kind of special area of the ribosomal gene. Really only AM fungal people look at this particular target. It's a large ribosomal subunit. There's a lot of literature about which different primers to use. So without boring people about bioinformatics, we use the LROR, and FLR2 primers. So we targeted that region right there in the middle. All right. So talk a little bit about the result of this first part, seeing how the AM fungal community changed. So what you're seeing here is the plant phylogeny on the right. So all these results are going to be organized by the plant phylogeny. And then across from left to right, there are going to be bars looking at the relative abundance changes in each of these AM fungal species, right? And then at the very top, you'll see the significance of whether or not that phylogenic effect we detected was a significant effect. So, a lot here, so I'll walk you through it. The first column is our most abundant AM fungal species, which was Anthropospora infrequens. It also, based on prior experiments, tends to be one that late successional plants respond very well to. So there's a gradient here from left to right of AM fungal species that we have detected larger level of growth promotion in late successionals. And so it's kind of organized in that range. So the most beneficial species are on the left and the least beneficial species are on the right. And we did detect a phylogenic effect in both years for our most common beneficial species. Interestingly, also detected a phylogenic effect of species that we predict to be non-beneficial. These are it's hard to say that they'd be detrimental, except in a competition scenario, right? They're just less beneficial to AM fungal species. But we saw a positive effect in year 2 for Rhamnocentra fulgida, and then in year 1 in Centrospora. So we saw it detected in 3 different species that the effect of how related the plant species were was significantly predicting the actual assemblages that we saw. Very cool. Overall, kind of community metrics, our Shannon's diversity was also predicted in year 1 by how related the plants were. So how diverse your soil community was kind of dependent on your phylogenetic position in the tree. Very interesting. Also density. So different parts of the plant tree had similar levels of overall AM fungal density. Very cool. So this is a pretty novel result because it's hard to test across this whole phylogeny. So we're very excited to be able to actually demonstrate that this phylogenic effect is happening. And then secondly, we're looking at plant life history, we also detected an effect of these life history traits. So again, this isn't as surprising because there's been previous research showing that late successionals are more responsive to AM fungi, but it was good to reproduce this result. F. mossi and C. chloroidium both predicted to be beneficial to late successional species. So it was really cool to see that they're growing better when they're growing with the late succession. So kind of intuitive, right? They benefit late successionals. They're growing better at late successionals. Interestingly, Rhamnocentra fulgida also grew better in late successionals, not the species that we detected any benefit to late successionals for. So it's kind of a neutral species, which is also being upregulated by the late successionals. Creates an interesting scenario to kind of test what might be the ultimate outcomes if you put these in a full feedback experiment and actually tested what are the reciprocal effects on the plant community. There are reasons to think that's going to be complex. So spoiler, we went ahead and tested it, and we'll talk about the results next. But yeah, so this first step still sets up this expectation that these feedback effects are going to be complex. And then again, alpha diversity, late successional host plants created a more diverse soil community in year 2. And then they also created a more abundant AM fungal community. So there was more AM fungi in general when you were growing in late successions. So all really interesting. I want to get to the ultimate conclusion though, of like, what does this mean for plant fitness overall? So to do that, we now have to do a second experiment. That second experiment uses this plant soil feedback framework. This is a framework it was published all the way back in '92 by my advisor, my PhD advisor, Jim Beaver. And it's a really clever way of dealing with these complex interaction feedback networks. So you think about these as pairwise interactions, right? So you got one plant species, they're competing, and they both have a soil community that's associated with them as well. And that's the whole, like the framework that we're starting with. And then we're talking about the fitness relationships between these different actors. So you have plant A, the soil community alpha, plant B, soil community beta. There's a fitness effect on the plants to the soil community. So right, they promote a soil community. And then alternatively, they have some sort of fitness effect if they were being planted in the neighboring soil community, right? So they have an effect on the fitnesses of the microbes that are most associated with them and with the microbes that are most associated with a competitor. Those microbes have fitness effects back up to the plants that they're associated with. They would also have fitness effects back up to the, right? This is a symmetric relationship where the soil communities that are promoted most by certain plants also benefit that plant the most. In this scenario, you expect kind of positive reinforcement, positive feedback. A lot of research in community ecology will tell you a positive feedback relationship is going to produce a less diverse community, right? There's reinforcement mechanisms. Generally speaking, the best competitor is going to win and just kind of take over the whole population. There's another scenario that I want to examine using this framework, which is a non-symmetric scenario. So in this case, we're still assuming plant A promotes the soil community alpha the most. Same thing with plant B and soil community beta. So these are still soil communities that are associated with that host plant, right? They are growing well with that plant. But they're actually giving really strong benefits to the competing species, right? So like it grows really well on plant. So soil community alpha grows really well on plant A, but if you plant plant B into that soil, it does really well. So in this scenario, you actually end up with an expectation of negative feedback. As plant A gets more populous in the community, it's changing the soil community in a way that actually increases the fitness of plant B. Again, in community ecology, a negative feedback, it tends to be stabilizing. So you could predict that this would be more likely to promote coexistence of two species that are directly competing. So now with just a mutualist, no pathogens, no other kind of microbes in the system, you can predict, you can still get positive or negative feedbacks in this system. So this framework is really nice because you can take this theoretical scenario and create an empirical equation to test. So you just basically take this two-phase experimental design. We did phase one. Now we're going to do the feedback experiments where we take the soil community we just differentiated and plant all the same plant communities back into that soil and test how big the plants grow. That's what all of those F terms are. It's the fitness, in our case, that is biomass of all these different plants and all these different crosses. Gives you a single number, interaction coefficient. That number is positive, positive feedback. That number is negative, negative feedback. It's a really clever system to get this really complex system of like interactions down to pairwise test, and you can get a single number that tells you whether or not you predict positive or negative feedback. So all the results I'm going to show you are going to be results of that interaction term. So that is a term that's calculated between the growth patterns of 4 different plants. So just keep that in mind. This is like a summary of the growth output of 4 plants to get you one number. This is just rehashing what I just said. But again, in phase 2, we're taking the soil communities that we differentiated in phase 1 and planting all the same species back into them in all the crosses. Ideally in all the crosses, I should say. 38 plant species, replicates of each of those plant species. You do all those crosses. I didn't have a big enough greenhouse, so I couldn't do all the crosses. But again, this experiment's really interested in the phylogenetic effect and the life history effect. So we can prioritize crosses that allow us to test those 2 effects. And that's what we did. So this is the actual experimental design. You don't have to like dig in deep into all the numbers, but keep in mind that we try to target across the phylogeny, make sure we hit early and late successionals. That way that when we go back and calculate these interaction terms, we have enough sampling between early and lates, and different areas and different branches on the phylogenetic tree testing both these effects. So these are the results. We did see positive feedback. So remember, interaction coefficient, positive number, positive feedback. We saw it when we had pairings of early species and early and late species. But very interestingly, when you pair a late and a late, you don't get a significant, on average, significant positive or negative feedback effect. It kind of is neutral. It's really interesting, and we'll talk a little bit more about what I think that might be some of the consequences of that result. We also did detect a phylogenetic effect as well. So it's a little interesting in this case. So none of these, except for the legume crosses, are actually significant. You can see the actual numbers all over, right? You get sometimes where the crosses are positive, sometimes with a negative. Mean effect, none of them were significant, except for legumes, which legumes have a positive feedback on each other, which is interesting. Again, they partner with rhizobia, so they have extra important relationships with the soil community. So it makes some sense that they would be a special case. And sure enough, they have a positive feedback when they're pairing with each other. And again, I looked at these distances between a phylogenetic tree. So that can be a linear predictor of relatedness. So I also looked at it that way. And here's where we got really stumped. This is phylogenetic distance. So a small number means that they're really close on the phylogenetic tree, which means they're really close relatives. A big number means they're really far on the tree, they're less related. And it is not a linear relationship, which was very interesting. It means this relationship is very complicated. One thing to keep in mind is that we're dealing with the whole plant phylogeny. So these really large distances are basically monocots and dicots. So as far away as you can get on the tree. So when you get that far away, you get either neutral or slightly negative feedbacks between these species. And when you're really, really close together, you also get either neutral or negative. But you can most likely get positive feedbacks between these intermediate relationships, which is interesting. We haven't fully teased that out yet, but like it's more complicated than a lot of people have thought this relationship to be. Like this fact that it's a non-linear relationship, these middle values of relatedness tend to give you the positive feedback is an interesting result. Again, probably because people don't necessarily always test across the whole phylogenetic tree of plants. So, the end result here is that we have observed feedback effects between early, and late. There's a complex relationship with relatedness, so it's non-linear. And most importantly, this different effects and feedback effects within life history categories gives you this kind of expectation for outcomes of competition. Namely, you might expect there might be an internal stable state in these systems because positive feedback effects create a reinforcement mechanism, right? You have a positive feedback effect, those type communities tend to reinforce themselves. The one kind of community that didn't have that positive reinforcement effect was the lates. So if you just have late successions together, they don't experience that positive feedback. So in these restored systems, you're getting stuck in this energetic level of this positive feedback between the early and the lates, but you don't get that with the late successional. So they can't drive themselves the system can't drive itself by itself all the way back to this late system. This is important for conservation because there might be a way to overcome that, and that could be soil amendments, right? So a big part of the fitness of these late successionals is the soil community, particularly in the prairie, it's the AM fungi. So if you can help push those soil communities back to a more late successional system, you might be able to overcome that positive feedback that these mixed systems are having. So not surprisingly, the people who study AMF say add AMF to the system, but it was an interesting result. So I'm going to change text here, but I just want to pause on this because I do think it is interesting that this is a good example of the system where understanding the microbes gives you an understanding that you just don't have if you just look at the plant communities. We all love the plant communities. We all really focus on them, but the microbes are playing an important role. And that's a good example of how ignoring these guys might be to the detriment of our endeavors, particularly in the restoration. All right, so now changing tacks to some newer work. I am, again, a microbe guy. I like microbes. I'm interested in microbes, but we also are interested in different ecosystems that they're operating in. And I'm going to talk about intermittent streams. So why am I talking about intermittent streams? So this work is part of a large EPSCoR grant called Aquatic Intermittency Effects on Microbiomes and Streams, AIMS. And the overarching goal of this project is to understand just how microbes are affecting biogeochemistry, hydrology, all these different things in these intermittent systems. And again, why intermittent systems? Well, these drying patterns make them hard to study. So I don't know, that's the next slide. No, I'll go into that later, but I'll just spoil it now. They're hard to study. Most stream ecologies deal with perennial systems. When there's no water in the stream ecologists kind of throw up their hands like, what do I do? So you need new techniques to understand intermittent systems. But they're very prevalent, and they're going to be more prevalent due to climate change, right? As we get more arid, as ecosystems become more arid, these intermittent systems are become more common, and we just don't understand them as well as we do the perennial systems. Why care about the microbes? Well, again, I'm a microbe guy. I can talk to you at length about why they're awesome and they're just inherently interesting. However, in stream systems, they're very important because they form the basis of the food web. The producers are microbes, and everything kind of just comes up from them. They also just represent the overwhelming diversity of life. So just in like a natural history kind of point of view, not understanding microbes, you're not understanding a huge part of the tree of life. Understanding microbes, understanding communities and streams in general is not a new thing. So there are a lot of frameworks you can lean upon to kind of help you anchor your thinking when you're dealing with these with stream systems. And the most venerable, the very famous, very controversial river continuum concept is one of them. So if you're not as familiar with this, it's a general framework that looks at rivers as this continuum of small zero or first order streams at headwaters that go all the way down to these large perennial streams at the higher order. And the river continuum concept, to grossly oversimplify, assumes that up at the top, you're going to get systems that are dominated by heterotrophs, right? So most of the production is actually happening on the landscape, and then you're getting all kinds of critters that eat all that inputs from the terrestrial landscape, and that's the dominant form of carbohydrates that are coming into the system. As you move down into the higher order streams, you get much more open water, much less tree cover, much fewer relative proportion of terrestrial inputs. Now you're getting autotrophs producing a lot of the carbon in the system. So you get these change in the shift in the community from the headwaters to the main channels based on where the energy is coming from. I also want to lean on a newer framework here for this analysis, which is the residence time hypothesis. So this, again, another hypothesis of how these communities might be structured in a stream, but this one deals a lot more with the idea of like how long has that water been in the network. The expectations here are about diversity, and basically, as in the high water in the headwaters in these low water streams, you're not only getting terrestrial carbon, you're getting terrestrial organisms. So a lot of runoff of organisms that are terrestrial in nature, which will inflate your diversity, right? There's a lot more taxa there because you're getting a lot of these terrestrial inputs. By the time you get all the way down to these higher order channels, selection has removed a lot of those terrestrial systems. And now you're just dealing with the aquatic specialists, or at least the higher proportion of aquatic specialists. So you should expect your diversity to go down because their terrestrial taxa have been selected. So this sets up our kind of expectations of just how the stream network is going to shape the microbial community. That's important because if we want to understand the microbial community, just the shape of the network is part of that equation. But this is an intermittent system. This is not enough to understand an intermittent system. You also need to understand how that intermittency is shaping the microbial communities. And that's one area where people are still trying to figure out what is the best way to do this, right? It's not as well trodden as like the shape of the network. And so, again, this is a map of contiguous United States and just the proportion of intermittent networks. You can see they're over 50% and they're just going to grow. So again, very important to understand these systems and that we just don't have a good grasp on them yet. So I want to understand the impact of hydrological connectivity on these communities. And to do that, I need, and there's a setup of these hypotheses now because of those frameworks that we're relying on. Higher hydrologic connectivity in one location should create higher diversity and abundance, right? So this is kind of extrapolating from those river morphology metrics and understanding, well, just longer presence of water, what is that going to do? We hypothesize it's going to do a similar thing where like water's been more present for longer, higher diversity, more connections to the terrestrial system, and higher density, more carbon coming in from the terrestrial system. Again, this is a hydrologic metric. We're talking about water in the channel. So I have some hypotheses about which of the different communities in the stream are going to be affected strongly. We tested in different compartments. And so my hypothesis is in the water column, it's going to be most sensitive to these hydrologic connectivity. Biofilms interact quite tightly with the water column, so we expect those to also be sensitive. Sediments and leaves tend to have a little bit more of a microenvironment, so they might be less sensitive. That's the hypothesis. Okay, so now we get to the challenge. How on earth do you quantify and test for this continuity? This is the thing that people are struggling with. So what we did is we used these relatively ineffective sensors that test, they're basically connectivity sensors, right? And you can distribute them throughout the entire network. In our stream system, we will put out 50. And they just tell you whether or not you have water presence absence. That's basically what you can get out of these things. They're really nice because they're cheap, relatively rugged, but that's about what you can get out of them, is whether or not there's water there. And then real-time sensors, they're recorded for 15 minutes. So you get really good data density off these sensors. But you need to take these water presence absence 15-minute intervals and turn them into a metric that's useful to understanding community's composition in a network. So how do you do that? Lots of ways. We chose this relatively new metric called communication distance, published in 2023. Without boring you guys on how this is calculated, you take presence absence, you turn that into a probability that there's water in a channel. I did this over a day, so like daily average. And then you multiply the actual stream length by that probability. So you end up with a length that gets bigger as things get more intermittent. You can think of it almost as a resistance to transport. The bigger the number, the harder it is for things to move by the channel, by our water transport. So that's our metric of discontinuity. Just real quick, because I wanted to do this metric at every point in the network. I did a sum, so cumulative value, and that number has to be normalized over area in order to, so it doesn't continuously grow. So we did cumulative communication distance by contributing basin area. That's the journey. Again, we did next sequencing to understand how the communities are changing. In this case, we targeted the 16S ribosomal region, which it's more standard, so I don't have a picture of what that is. But like, it's again, Illumina sequencing, we're doing metabar coding. We also verified and then did transfers. Yeah, if anyone here is a bioinformatics nerd, I'm happy to talk about it, but I'm assuming most of you don't want me to dive into those details. All right, so we're going to look at multiple linear regressions. The terms I included are this cumulative communication distance is our intermittency metric. Also, stream order is this classic kind of stream morphology metric. Also, these sites that sometimes go dry, that's going to be an important predictor. So we also included a binary wet-dry predictor as well. Importantly, the microhabitat just ended up being the largest determinant of community composition, but they're just very different, which is not surprising. Leaf communities are very different than water communities, which are very different than biofilm communities. To account for that, we just ended up running separate models for each of the compartments. So it's 4 different analyses for each of those compartments. So going into the results, this is stream order, right? So this is kind of getting at the stream morphology. It's kind of classic metrics of how do you understand how the community is being structured. Interestingly, the one that got predicted in terms of alpha diversity was leaf community, which stumped me because I did not predict that at all. So this is kind of counterintuitive to my hypotheses. I thought the water community would be the strongest to be predicted by hydrologic connectivity, followed by the biofilms. Everything grayed out, it's just not significant relationship. The only one that was significant in terms of stream order was the leaf community. The first order streams had the highest diversity, which you might expect this corresponds to river continuum concept. Except these systems are prairie streams, which means a big part of the river continuum concept is that the headwaters are forested, a lot of terrestrial input, a lot of leaf input. It's the exact opposite in this system. All the headwaters are in the prairie, and you only get these forested systems down in the middle of the channel. So it's kind of inverted of like the classic system. So we're still seeing the effect, but it kind of shows us that this classic conception of like, well, it's these forested headwaters and then it's these open channels in the high order streams doesn't really explain this pattern, even though that's the traditional way of understanding what's driving river continuum concept. So in this prairie system, we see the same pattern, but it's being driven by something else. Again, this is now intermittency. So this is the thing we're trying to understand that's novel. Leaf area, again, was the only thing that diversity was explained by. In this case, higher numbers means more intermittency. So the more intermittent the system, the higher the alpha diversity in leaf packs. Interesting. I would say I don't have a strong explanation of this because I didn't really expect to see it in the leaf packs. But you can imagine the leaf packs wetting and drying, that's creating a more diverse community there. That could help explain it. Also, leaf packs tend to kind of hold on to moisture. So in a system dries, it tend to be one of the last places to fully dry. So that might help explain why they're seeing a pattern here. But again, the inverse argument is also valid, right? Like, why is the biofilms that completely dry not showing a strong impact of intermittency? Can't explain that. So it's very interesting. This is clearly just evidence that this is a complicated story, and we still have a lot to understand. However, intermittency does predict alpha diversity in leaf packs, which is interesting. Now, the most important predictor definitely ended up being flow state. So is the site wet or dry, period, was the biggest predictor. Very interestingly for me, the one case where the diversity was not predictive was sediments. Again, water's not here because you can't sample water when it's dry. So we're only looking at these three compartments. But sediments, I would have thought would have been the most sensitive to this. And they were not sensitive at all. The diversity of the soil communities did not change whether the system was wet or whether the site was wet or dry. Moving on to the next set of results can be even more interesting, but like just keep a pin in that. So now we're looking at alpha diversity. The other thing we want to look at is overall density. So in this experiment, we were blessed enough to be able to do qPCR. We get you a very reliable metric of like the actual abundances of your microbes. And we saw an effect, but not in leaf packs. So now when we're looking at just the abundances, we see it in biofilms. This one, I understand. So classically speaking, you get to a third order stream. These are the wider open streams. In theory, they should be getting more sunlight. Again, the fact that this is an inverted system where the third order streams have the most tree cover kind of makes me scratch my head a little bit. But in the theory, these wider order streams should get more sunlight, more autotrophic. Biofilms are highly autotrophic systems. So that could make sense of why the biofilms are having higher abundances in these higher order streams. Now, the sediments show the inverse relationship to intermittency when it comes to these abundances, excuse me. So the sediments, when it's more, when there's more intermittency, you get higher abundances in the sediments. Now, that might sound counterintuitive until you also look at the effect of wet dry, and it kind of reinforces the same thing. Dry sites had higher abundances. Not what I expected. And we scratched our heads a long time to try to figure this out. And we came up with a reasonable hypothesis I think is right, which is sediments, when they become inundated, are very quick to turn anoxic. So we might be looking at the effect of anoxia here. This is a really cool theory. Unfortunately, we didn't think about it beforehand, so we didn't put oxygen sensors in sediments. So it's hard to prove that that's what the story here is, the oxygen story. But I think it's a pretty good hypothesis. And if I were to rerun the experiment, I would definitely have tested the soil oxygen to kind of confirm. But I think that's what's happening here. All right. So for our conclusions of this experiment, all three metrics ended up being useful in different compartments in different scenarios. So it's not I'm not going to turn over the apple cart and completely rediscover how we understand these communities and the stream systems. All the classic systems still help. But it's a combination of all three. Like, and in these intermittent systems, discontinuity, like whether or not, how often the system's going dry and wet, that ends up being important to understand these communities as well. So it's an all of the above approach in terms of understanding these communities. And the binary wet dry ended up being really, really important. And I think that's an oxygen story. I wish I could prove it. But I think it holds up in terms of conceptually. All right. Now, one last shift here, which is what I'm going to do here now in ESIIL. So I've talked a lot about microbes. It's my passion, basically because I think they're a really cool study organism. I think the fact that you can get really dense community data from a very small sample, the richness of bioinformatic data makes them a really interesting target of dynamics, and I'm really interested in community dynamics, mutualisms, especially, but like all community dynamics. So I love working with microbes, and I want to keep doing that. So the project I'm doing here at ESIIL, again, is niche modeling, trying to understand the spatial distribution of these guys. And I want to understand a mutualistic system. So I want to understand both of the organisms at the same time. So in my case, I'm going to be looking at plants and fungi, because I know a lot about them, and I'm hoping to model them both at the same time. This is my current data set. My original plan was to do the contiguous United States. If you look up here, you can kind of see that I didn't quite get full coverage of the US. So I think I'm going to scale back a little bit. What I do have a pretty good coverage of is this is pretty much the prairie right there, like the Prairie Peninsula way over here, getting close to Illinois. I think I got a pretty good coverage of the North American prairie, so I want to do the same analysis, but maybe limiting myself to the prairie system, the system I know the best, honestly. And then if it goes well, maybe like write a grant, see if I can get some sequencing of like neon or something like that to get a better coverage of the whole US. But I think this is a good place to start. And again, I know AMF very well. I know prairies really very well. So I'm hoping that background knowledge is going to help me kind of get this project off the ground. Also had tons of side projects if you guys want to talk about them. In terms of niche modeling, this original idea to study mutualisms started from a data set from 2015 that I still haven't analyzed between heliconia plants and hummingbirds. That I have a advisor that is, or not advisor, a collaborator who is desperate for me to get back to. So that would be a fun side project. I do still like working with soil feedback systems. I think that soil feedback framework is a very useful system because you can get those interaction terms. That's a really good way to simplify a really complex problem. And yeah, I'm a microbe guy, so metabarcoding all applies. If you have any metabarcoding questions, just ask me. And with that, I'll leave it up for questions.
I'm curious about your second project. When you say dry sediment, are we talking like sand, mud, like?
These were all fairly well-established streams. So it was a stream bed. You can tell the
Yeah.
difference between the stream bed and the banks, but it
no,
was,
like flow in the stream, but it's not like baked.
no, it's baked.
It's baked.
It's
Okay.
baked. It's one of those systems where like you can tell there's a stream bed. It is different than the banks, but there are parts of the year where it is bone dry.
Yeah. Yeah, I mean, that's what I was curious is like where your sensor is cutting off really dry. And then I guess kind of a follow-up on that, I'm curious if you have, and presumably not you, but if other folks have taken data on the hyporheic flow around there, and if, do you think that may have an impact?
It definitely has an impact. I think every hydrologist on that project I've talked to has been like, I wish we had sampled more in the hyporheic zone, but they did have wells. So they had a well that was right at the surface and a well that I think went 2 meters down or a meter down. I can't remember exactly. So they were able to sample there. I thought that would have been good enough, but all the hydrologists were like, no, we need to go deeper than 2 meters. That's not deep enough to really understand what's going on. So like we have that well data. I haven't done that much in the well data. They didn't sequence the well data, so the microbe guy kind of got left out of that analysis. But they are trying to understand that because it almost certainly is having an effect, especially. So this is across 3 different EPSCoR states. So there's sites in Idaho, there's sites in Kansas where I was working, and there's sites in Alabama. And the Kansas sites, particularly Kanza, which you saw nice pictures of throughout this, the geology there is insane. Like it's these layers of shale and limestone that just creates really weird like drainage patterns. That system that I showed data from is a system where it kind of dries from the outside in. So like the bottom of the watershed will dry and the top of the watershed will dry, but it's the middle that stays wet. That's all due to springs. So like there's a lot of underground water flow paths that's really important there that I'm not a hydrologist, but talking to enough of them, that's a whole thing unto itself of just trying to understand, even in just that one site, let alone across all the sites of the experiment. So I wish I had a better answer other than yes, it's important and we're trying to figure it out.
Yeah. Yeah, I mean, I just know enough to be dangerous about it, but
Yeah.
but yeah, it seems like thinking of the stream as having like a real distinct boundary is like a little misleading and maybe related to some of those.
The thing it was hammering into my brain on this project was the saying, a stream is just where groundwater ran out of ground. And it's very true. Like the groundwater behavior is very important to these systems. Now, with the microbes in my little tiny world, the microbes do have like this special importance with surface water flow, right? Like they do tend to transport it through surface water. There's some evidence of things happening with microbes in the hyporheic zone, but it almost certainly is a completely different kind of community down there than it is in the surface water. So if you're interested in the communities shaped by the surface water, you can kind of cheat and make that distinction. But no, agreed, especially because this project was looking at biogeochemistry. it's really hard to ignore what's happening in the hyperemic zone and really fully understand what's happening with biogeochemistry. So, well, point well taken. Yes.
I have a question about the alpha diversity on the leaf litter.
Yes.
I guess I was wondering if you think that just like the substrate quality of the leaves, or like how connected the substrate is compared to like it being more dispersed in the sediments or in the water, has an effect on the microbials community, like ability to evolve and diversify?
I think I will cheat again and say, of course, definitely, but we didn't necessarily have the data to solve that. So in terms of like the actual like structure of leaf packs, that would have been really difficult to analyze, but almost certainly was important, right? Like, is it a small pack that's isolated, or is it like a long sinuous patch? All that probably had a big effect. We didn't characterize those leaf packs spatially enough to really be able to get at that, let alone like know what method to use to analyze it, but almost certainly that matters. We did try to get at a simpler metric of like quality of the leaf packs by understanding what species we were sampling from. We always try to take the leaf from whatever the most common tree was in that area, and we tried our best to record it. This was a big project with a lot of people, and not all of us knew our trees very well. And so that data ended up being kind of messy, the actual species data. And so that became difficult to analyze. Like, in an ideal world, we would have had a much better species identification of the trees. And that litter quality would have definitely have been a really strong predictor of these communities. We still have that data, and I still want to look at that data, but it is just really messy because there was vastly different levels of skill at the people who were collecting this data in terms of identifying trees. Yes.
Just to follow up on that, so when you say leaf pack, is it just, you're not putting them out there? Because I've done experiments where you put out leaf packs. It's just whatever native.
It's what's native. We did do some other follow-up experiments out there where we were adding in leaves to do like decomp studies and stuff like that.
Yeah.
This sampling, it's all environmental sampling. So what leaf packs were there?
Okay.
There was a handful of times where we couldn't find any leaves, and that was documented. For the most part, though, there were leaves present, so we were able to sample.
So even in the, as you said, like the headwaters were mostly grassland.
Yeah, sometimes they sampled grass, and then sometimes they didn't, and that was not consistent, unfortunately. But like even up there were usually at least a few leaves that we were able to find.
Like deciduous leaves?
Yes.
Okay.
Again, there are trees on the landscape, even though it's a prairie, and like, there's a lot of overland flow, so things do wash in, and everything tends to settle in the stream bed. But like definitely it was harder picking up at the top, and sometimes we just didn't have a leaf sample for a site, and we just dealt with that zero.
Interesting.
Looks like we have a question online from Ty. Ty, if you want to unmute and ask your question.
I do. Thanks for a great talk. So I couldn't be there in person, but I really enjoyed a lot of the things. I think we have a couple months of good discussion about all the things that you talked about. But I think I'll start with one that dispersal really stood out in my mind in this story, that as a possible mechanism, but not something that you tested a lot. And sort of the setup, I guess, for that, for my question would be, in the theory world, there is a lot of work in sort of a species agnostic context about just the networks of rivers, and that a population at the headwaters is experiencing a lot of competition with its environment, and a population in the sort of open big waters is experiencing a lot of biological competition. And so in these ephemeral streams, water might just be a dispersal mechanism. So you're getting microbes competing with their environment when it's dry, and then you get water moving in, and it's just a dispersal mechanism that allows for a lot of biological competition.
Yep.
Anyways, my question is just about the role of those two mechanisms, competition and dispersal, in the river network and in the sort of intermittency. How could those be playing a role, and how important do you think they are?
I think they're super, it's a great question. I think they're super important. I'm only just starting to get my head around how to test that. And like, so I've been reading a lot of like meta-ecology literature, trying to get an idea of like what kind of analysis framework to use to really get at that question. But yeah, no, it's huge, right? And I think a big part of the story that this current analysis doesn't get at is the fact that as things are drying down, as the intermittency goes up in the parlance of this analysis, you're getting more local control. It is much more like site by site. Things are just site specific. And so like if you were to plot the variance, which I didn't show in this presentation, but the variance skyrockets in the communities as you get as it shows you right now, because it's much more site-specific controls. And then as things connect, it's much more just like you're saying, it's much more about distribution between the network, right? So things become more similar as things connect, and they're much more dissimilar as things disconnect. How to parse that out is an analysis problem that I am still kind of getting my teeth into, but I think it's an important part of this story because I just anecdotally, just by looking at the data without running an analysis, you can tell variances are skyrocketing as things dry up. So that's a huge part of this. Yes.
I have a question about the first research question and the way you set up the design. The plants that you use, were those seeds sourced from the prairies themselves?
As best we could. There were a couple things, particularly grasses, that we ended up having to get from a distributor. But you know, that the lab I worked in was blessed with a lot of really good prairie experts. So anything that we could go to the prairie and collect seed for, we did.
Right.
But I don't have the list with me, but if you're interested, I could get the list of which ones were actually prairie sourced from the prairie and which ones we had to get from a distributor.
And you didn't notice any major changes between, did you track like?
I didn't track that. That's a very good point, but that would be interesting to go back and see, like, is there just an effect of, well, which ones came from the prairie and which ones came from growers? I haven't checked them. That's something to check for sure.
Yeah, that would be interesting.
Like Ty has another question.
Oh, go ahead.
I have a question about the first experiment. You didn't talk much about herbivores. Now I know herbivores are a big player, not just because they chew on plants, because also they move around nutrients. But you sort of looked at these two plants competing in a vacuum without nutrients moving in and out. Just curious if you think it would change your results or affect your results at all if you did it in a more herbivore-rich experimental design.
I think so. I think without knowing for sure, my hypothesis would be the AM effects would just be stronger. One of the benefits that people have detected from AMF is just general improved resilience of plants. Often this is in pathogen resistance, but I could imagine the same effect being seen in herbivory. And again, the mechanism is you can kind of understand the mechanism. They're getting more nutrients from the AMF relationship, so they're able to respond better to stressors. But that's a miracle question that we didn't test, so it's hard to say. But my hypothesis would be that herbivory would increase the importance of these AM partners in the species that are responsive to them. And that would upregulate the need for the EMF in those species. And then the ones that aren't responsive, which tends to be the early successionals, that one's harder to predict. But yeah, I think definitely the late successionals, there would be an upregulation of the need for their partners.
Awesome. All right, thank you. Thank you, Rob.
Thank you.